Thank you so much for having me here today. It's a real pleasure to speak to you about different approaches where we may apply genomic and phenotypic data to rare species conservation and management, leveraging our conservation genetics toolkit. Before I get started, however, I do want to acknowledge that the Pennsylvania State Universities, where I'm coming from today, the campuses are located on the original homelands of the Erie, Haudenosaunee, Lenape, Shawnee, Susquehannock, and Wazazi nations. And so as a land-grant institution, we acknowledge and honor the traditional caretakers of these lands and strive to understand and model their responsible stewardship. We also acknowledge the longer history of these lands and our place in that history. In addition to the acknowledgement of land, I want to say before I get going that when thinking about rare species, the key to their conservation, I believe, is collaborative partnerships. And the information I'll be providing today really depends upon awesome collaborative partnerships across multiple institutions, and particularly the leadership and foresight of Dr. Tom Ledegg, who established many common garden experiments as part of his research program, collaborations with Jessica Wright from the U.S. Forest Service, and amazingly motivated PhD student, Dr. Lionel DeSanto, in addition to many, many other individuals that have been key to this research. So I want to acknowledge all of the participants and those that have provided funding to support this research. So today I wanted to talk about the genetic consequences of rarity. I'll focus on long-lived species, but you can imagine all species are going to be subject to substantial shifts in the environment, including fluctuations in both abiotic and biotic stresses that will impact population demography. And so if we think about a species' evolutionary potential, it's tightly linked to both the amount and distribution of genetic variation. Genetic variation is necessary for natural selection to act upon and is thus a prerequisite for adaptation. And as conditions change, the maintenance of that genetic variation becomes essential in maintaining a species' adaptive evolutionary potential. However, the maintenance of that genetic variation, particularly for our species of conservation concern, is a significant challenge where the combination of environmental changes, demographic changes, may impact the distribution of that genetic variation needed for long-term species persistence. So developing conservation management strategies for species requires this understanding of the interactions between environmental variation, population demography, and how it may impact the amount and distribution of genetic variation, and thus species' adaptive evolutionary potential. So the goal of my talk today is to discuss the importance of preserving existing variation and approaches to preserving existing variation, but also to ask the question, under what conditions might we need to supplement existing variation, and what data might support a plan for that supplementing existing variation, specifically using a genetic rescue approach. So how might genetic rescue impact a species' evolutionary potential? Where we're defining genetic rescue as natural or facilitated introduction of genetic variation from unrelated individuals with an aim to increase population fitness. So genetic rescue can be looked at in terms of interpopulation gene flow or even interspecies gene flow. And so we can imagine that we have a population of a particular size and it undergoes some environmental change. Following that change, there could be a potential population decline or maladaptation to those changing conditions results in extirpation. And once populations fall below a particular extinction threshold, they could potentially go extinct. Now, where genetic variation comes into play here is important. So if that population has enough standing genetic variation within populations, perhaps it could mitigate that population decline, evolve in response to those changing conditions, and undergo rescue known as an evolutionary rescue from that standing genetic variation within populations. However, one of the questions that we wanted to ask is, could we facilitate a rescue where the introduction of genetic variation from another population or even potentially another species could reduce the demographic consequences of maladaptation, increasing that rate where we might see populations evolve and increase population size over time. And so thinking about genetic rescue, we wanted to focus on the Torrey Pines species. So today I'm going to focus on Torrey pine as a case study because Torrey pine is kind of a poster child for rarity in forest trees. And we'll discuss the things that we knew going into a project where we were considering genetic rescue and the things that we've been learning. And so, as I said, Torrey pine, it's one of the rarest pine species in the world. It's restricted to just two populations. It has one mainland population just north of San Diego, near La Jolla on California, within California state parks, and it has an island population that is conserved by the National Park Service. And so the island population is on Santa Rosa Island, one of the Channel Islands, and it represents a population that is currently expanding. With the relatively recent removal of goats on the Channel Island, Torrey pine, which was once fairly restricted in its range on the island, is currently expanding. And we're seeing evidence that this population is marching across the island into new terrain. Indeed, this population is extremely interesting actually because it has a large number of mature trees, but also a good number of trees that range in age class. And this is really important because we see the distribution of age classes and we also see evidence for natural recruitment on the island population. Now, this contrasts with the mainland population, where there appears to be very limited natural recruitment and less diversity in the range of age classes observed than on the island population. Torrey Pines State Reserve is challenged in its geographic location, where it's surrounded by human development on all sides. And in addition, the populations at this site are currently being decimated by the five-spined engraver beetle. Despite extensive efforts to control the beetle population, a large proportion of trees on the mainland of similar age class have been lost due to beetle kill. So IUCN currently recognizes Torrey pine as critically endangered due to development, fire, climate change, drought, pests, and disease. In addition, previous genetic analysis based on allozymes and chloroplast sequencing indicate that these trees are nearly genetically identical. except for a very few fixed differences between the mainland and island trees. So previous genetic studies have seen no genetic variation within populations and only few differences between populations. This degree of monomorphism is virtually unknown for a conifer species and represents a substantial conservation concern. So the combination of susceptibility to environmental stresses, demographic stochasticity, and lack of genetic variation may impact Torrey Pines' persistence in situ and its future evolutionary potential, which is why we were interested in thinking about genetic rescue for this species. So how can genomic and phenotypic data inform our conservation strategies? Well, let's first talk about genomic data. Given that we had seen limited genetic differences between Torrey pine populations and no genetic variation within populations, we wanted to take a genomics approach. And one of the reasons that a genomics approach makes sense for Torrey pine is that pine genomes are really very large. If we look at this graph on the bottom, you can see the genome size is on our y-axis here, and we have the assembly quality here for references and draft genomes. And if you imagine, so our classic model for plant species is Arabidopsis thaliana. It has a genome size of about 135 megabase pairs. So pretty small genome, really high quality reference genome. And what we see is with a number of species increasing in genome size. We can look over here, we see our barley. It has a genome size of about 5.3 gigabases, which is pretty big. But conifers have genome sizes that are well over 10 gigabases. And this makes them a little challenging for multiple reasons. Therefore, to get a good understanding of genomic variation within a pine, taking a genomics approach is appropriate because it allows you to survey across the genome. So just based on genome size, a genomics approach provides a more fine scale approach to identifying genetic variation within your population. In addition, the advantage to taking a genomic approach is that you can test models that may indicate how different evolutionary processes have contributed to genetic structure over time. So this is increasingly relevant for species of conservation concern, because one of the questions we often ask with rare species is, has that species or population always had a small population size or small effective population size? Is there evidence of different patterns of colonization, founding events, bottlenecks that have influenced the genomic structure of populations? And combining genome-wide estimates of genetic variation using genomic data with simulations allows us to address these types of questions. Finally, genomic data allows us to identify loci that may be candidates for selection and important adaptation. Now, the advantage of taking the genomics approach, allows us to survey genomic variation across an individual's genome, where we might identify some genetic markers, some loci that are outliers, relative to what we'd expect based on neutral evolutionary processes. And so the idea behind these approaches outlined here below is that we can identify loci that are maybe more genetically different than we'd expect by chance. So here on the left, we have a number of different loci in black here, and they have a range and allele frequency from 0 to 1, and each of them has an estimate of genetic differentiation. And so this dashed line gives us an idea of the genome-wide average for genetic differentiation across all loci across the genome. And so what we can do is look for loci that are outlying relative to that genome-wide average. And these could be candidates that are under selection and potentially important to adaptation for our species of interest. There are multiple ways we can do this. We can also take distributions of genetic differentiation here, where we have our FST estimate, and look for those loci that are in the tails of the distribution that are maybe more different than we'd expect by chance over here, or more similar than we'd expect by chance over here. So taking these approaches to identify outlier loci are incredibly valuable because they maybe can help you develop hypotheses for potential adaptive genetic differentiation in your population or species of interest. In addition, phenotypic data can be extremely helpful to inform conservation strategies. And particularly, I want to focus here on the advantage of common garden experiments. The advantage of common garden experiments is that you can sample individuals or seeds from a variety of populations, different environments, and grow them all together in the same place, so that any differences that you observe within that same common environment are likely due to genetic differences. And so you can evaluate whether phenotypic differences have evolved across different populations or across populations associated with different environments. In addition, if you're developing a pedigreed program, you can maybe evaluate in that common garden environment if there are fitness differences between your pure species or hybrid species. If you have F1 crosses of a species and you're growing them alongside your parental populations, you can see if there are fitness differences in making those genetic mixes. And finally, where you have genomic data for individuals within your common garden experiment to pair with phenotypic data, you can associate genetic variation with phenotypic variation, and even maybe potentially test for barriers to reproduction that may evolve and impact genetic rescue recommendations. So the first question that we wanted to ask using genomic data for Torrey Pine is, how is genomic variation distributed within and among populations? We sampled Torrey Pine on the mainland and island, 140 individuals on the mainland, 130 on the island, and took our genomics approach. We used double digest restriction, digest sequencing, dd-rad sequencing, and with this sequencing, we were able to develop over 93,000 single nucleotide polymorphisms, or SNPs. And so the first question was to just look at genomic structure of our mainland and island populations. And to do this, we performed a principal components analysis using all of our genomic data, all of our over 93,000 SNPs. And the first thing that you will note is that there are differences clearly between our island population and our mainland population. Interestingly, So you'll note that PC1, only 1.9% of the variance is explained by this axis, and PC2, only 0.6% of the variation is explained. This suggests that between populations, there is still very limited genetic differences between these two populations. However, what was really exciting about this result is that previous studies have suggested there is no genetic variation within Torrey pine populations. And what our data here tell us is that across this PC2 axis, we can see that there is variability within the populations. There's not a lot of variability, but it does exist, which is really exciting. Overall, we can compare our PC axis with our measure of genetic differentiation, FST, which is also really low. FST ranges from 0 to 1, so a value of 0.013 is quite low, suggesting there are very limited genetic differences between the populations, and our estimates of heterozygosity for both the populations, again, were quite low, particularly for a conifer, where we compared these values to other conifer species, and we see that estimates of heterozygosity are quite low. So it confirms previous observations suggesting that there are genetic differences between the populations, but there are only few genetic differences. And it counters previous observations suggesting that there is, however, variation within populations, which is very exciting and important in the consideration of genetic rescue. So the next question that we wanted to ask is, how have different evolutionary processes contributed to genetic structure over time? We really want to understand, has Torrey pine been affected by bottlenecks, colonization routes? What processes has influenced the genetic structure of these populations, and is it changing over time? And to do this, we take a simulation approach. And so we can develop several models that might indicate how Torrey pine contemporary populations have formed. One of the models we tested was that there was a common ancestral population. Over time, it diverged into an island and mainland population, and that there was some recurrent, very limited, but some recurrent gene flow between the two populations. Another type of model that we tested within this scenario was that the mainland population was the ancestral population. It stayed kind of consistent population size over time. But at some point in time from the mainland, the island population was colonized and the island population has since expanded. So these are types of models that we can assess and compare for Torrey pine and for different species to understand demographic history. And we can also suggest that there has maybe been the island as the ancestral population and that the island colonized the mainland population. So these are just examples, but what we do is we test for which model has the most support. And what we found in the case of Torrey pine is that the model with the most support had an ancestral population that diverged into both the island and mainland population. And from these data, we can get an understanding of the approximate effective population size of our ancestral population noted up here, and an estimate of our contemporary effective population size of our island and our mainland population. We also can parameterize different components of the model. And what we noted was that there is some limited migration between the island and mainland populations, and this may be contributing to the fact that there is low genetic differences between the two populations. So overall, what does this tell us as conservation genetics approaches? So it suggests that we see low genetic diversity in both Torrey pine populations, supporting previous findings. But we're really excited that there is more genetic variation within the populations than was previously observed. Second, we see that there is limited genetic differences between the two populations, and this is supported by the fact from our demographic modeling and simulations, which suggests that there is some contemporary migration between these two populations that might be contributing to more homogenizing the populations. So these data overall provide a good description of genome-wide averages of genetic variation and differentiation between Torrey pine. But if one of our goals is to preserve adaptive evolutionary potential, we want to see if there may be regions of the genome that may be under selection and therefore important to adaptation within either the mainland or island environments. And so that again brings us back to the value of phenotyping, and particularly common garden experiments. So we were really lucky to benefit from a resource established by Dr. Tom Ledig, where he established a common garden experiment taking individuals from the mainland population, individuals from the island population, and also seedlings from an F1 generation representing island mother trees that had been pollinated from mainland pollen. And he planted them and grew them all up in one common garden experiment. And they had been phenotyped for a number of different characteristics, including height and reproductive activity. And so we wanted to see if any phenotypic differences had potentially evolved between our two populations that might suggest adaptation to either of the island or mainland environments. And so, despite evidence for low genome-wide estimates of genomic differentiation between populations, what we see is multiple lines of evidence that suggest phenotypic differences between the populations exist. So for a first example, we have our common garden experiment, and we looked at height accumulation between the island, mainland, and F1 hybrids. And what we can clearly see is that relative to each other, F1s show greatest height accumulation over time. And there are clear differences between the height accumulated within the mainland populations and the island populations, pointing towards the fact that there may have been some evolved differences between these two for the phenotypic trait of height. In addition, previous research had seen that there is differentiation between the two populations for co-morphology, and we complemented this research taking X-rays of our ex situ seed collection from the island and mainland population, and we saw, based on a number of different seed characteristics that we evaluated as part of our X-ray seed morphology collection, we see that there are clear differences between the island and mainland population for seed characteristics. Interestingly, these ex situ conservation collections become really valuable because they provide a great way to have baseline data for potentially evaluating variation in your populations. So what we did with our ex situ collections is we collected seed in the field that had been identified by maternal family. So we know what population it came from and what mother the seed came from. And measuring all of our morphological traits, we divided up the phenotypic variation that we observed into either being explained by population differences, maternal family differences, or individual residual differences. And what you can see here is that clearly populations differed for a number of these traits, but also we see a surprising amount of variation is coming from maternal families. So this told us early on that there was probably maternal family structure within both of the populations. So taking a genomics approach should find within population genomic variation, which we did find. So this was a really good way to move forward on a genomics project because it told us that in advance, we should be finding genetic variation within populations if we used a genomic approach. So we see phenotypic differences between these two populations that suggest they may have evolved differences within either the mainland or island environment. But what we really want to do is take a genomics approach to this. Can we identify potential regions of the genome or candidate genes that may be under selection? And so going back to thinking about those outlier approaches I discussed earlier, we conducted 3 different genome scans to analyze the whole genome to identify those candidate SNPs that might be outlying relative to genome averages, reflecting potential candidates that might be important for adaptation. And so we chose 3 different programs to do this. There are a number of different programs that do this. We chose Bayescan, PC Adapt, and Outflank. And the reason that we chose 3 programs is because where we find commonality across these programs, we can be increasingly confident that these are candidate genes that might be important to selection. And overall, we find over 2,000 SNPs that are potential candidates for selection. So as we go further down this path, we can understand what these candidate SNPs are involved in from a functional standpoint and relate that understanding of gene function to phenotypic differences that we see in the field. So what does this tell us? Going back to this figure, we've got low genetic diversity, limited demographic expansion, and limited genetic differences from a genome-wide perspective. However, our phenotypic data suggests that the environments may not be equivalent and that phenotypic differences may have evolved between the 2 populations, and that we've identified potential candidate SNPs that could be important for adaptation between the 2 populations. So overall, incorporating this knowledge into a Torrey Pine conservation strategy, how do we do this? Well, we've got our genome-wide survey of genomic variation that says there's limited genetic variation within and limited differentiation between, but there are differences within which could be important to implementing a genetic rescue program. From a demographic perspective, we see that the effective population size has historically been low. There has been some very limited expansion, and there is evidence that there is some contemporary gene flow between these populations. If we're going to do another ex situ collection, If we want to sample neutral standing genetic variation, it may be that we only need to sample one population. However, what does this mean for adaptive variation? Well, we've seen evidence of both phenotypic and genomic data that suggests there is candidates for adaptive genetic differences that could be contributing to some of the phenotypic differences that we've been seeing between the two populations. So if we want to make recommendations for ex situ collections that target sampling both neutral and adaptive genetic variation, we recommend resampling both populations to capture that adaptive genetic variation. In addition, we've seen some evidence that there is a fitness advantage in the F1 in our common garden experiments. So it will be really important to look at the fitness consequences following admixture across multiple generations in a common garden experiment, including F2s, back crosses, and beyond, to see if there is any change in fitness following additional pedigrees to these Torrey pine populations. So overall, what does this say to monitoring genomic diversity? Well, if we see that Torrey pine provides a valuable case study, I think, for the contribution of genomic data to conservation management strategies. We can use genomic data to evaluate the distribution of both neutral and adaptive genetic variation. We can use genomic data to get a better understanding of species evolutionary history, and that can help us optimize our conservation management strategies. In addition, common garden experiments, where possible, permit the evaluation of phenotypic differences among populations that might be important to adaptation, and understanding the evolution of adaptive differences can help us evaluate risks associated with implementing genetic rescue designs. And so we know that IUCN has set ambitious goals with regards to safeguarding species diversity, protecting ecosystems, and preserving habitat. However, it's clear that intraspecific and interspecific genetic variation remains a gap in these strategies that needs to be filled. And this will require partnerships between governmental, non-governmental, industry, and academic institutions to make genetic data practical, affordable, and accessible. And there are still some challenges to overcome, but I'm really excited for meetings like these, where we see that the capacity is there, and we are building on that capacity to address these challenges. And with that, I'm happy to take any questions that you might have. Thank you so much.